Hello, everybody, and welcome back to episode number 29 of the Roses Are in Rhetoric podcast. As promised last week, we are joined today by Dr. Guy Bouvier, a neuroscience researcher at the University of California, San Francisco. Guy, thank you for joining us.
And thank you so much for inviting me, guys.
Yeah, we have a lot of fun in this episode. We're going to kind of cover three broad topics. We're going to first take a look at the specifics of Guy's research. Then we're going to kind of get a little more broad. We're going to take a look at research in general. And then another topic that Joe and I like to incorporate into the show as well is to try to understand how these different areas, whether it be research or other areas of interest, affect the people on a personal level. And so we're going to get to that a little bit later on, plus an album of the week and our new segment, our social media shoutouts. But before kind of going into that, Joe had kind of a fun idea. Guy, we were both given a paper by our mutual friend Cameron Wollheit that you had published discussing kind of the visual cortex and the way that it incorporates motion and luminosity. And Joe's idea was that him and I would each try to explain to you what we think your paper is about, and you tell us how far off the mark we are from whether or not we understood it well. So this should be a lot of fun. I'm going to pass it off to my charming co-host, Joseph Stanford, to start things off. Joe, when you're ready, your take on the paper from Guy Bouvier.
Okay, perfect. So the paper, as I understood it, the research was an investigation on the effects of head position on the brain, as well as the effects of light on the brain's registration of those positions. So I understand that the brain has like a type of system in it, similar to your cell phone.
First question, should I interrupt you when something is not necessarily accurate? Or
No, just save it for the end. Save the ridiculous. I'll get there. But
overall, it's great. It's great. But
okay.
Go ahead.
Okay, so I understand it to be like there's an accelerometer in your brain, kind of similar to your phone, like the device in your phone that tells it which way is up.
Yeah.
And that same sensor exists within the brain, and that's what you were studying via mice. And there were certain things that had effects on that registration of the accelerometer for your brain.
Mm-hmm.
And some of those things are changes in light intensity, and then some other things that you investigated. So that's my understanding of the gist of the paper. Jim, do you want to explain what your take was?
Luckily, or perhaps unluckily, I had a similar take on the paper. So we'll have to see what we have wrong. The only thing I was going to add to it is kind of there's a group of cells in the brain that Guy refers to as SOM5 cells. And the way that I kind of understood these cells working, Guy, is that they kind of integrate the light information with motion information. And when it's dark and your brain is sensing movement, your brain kind of lowers the signal intensity of the visual cortex, in a sense because it doesn't really trust what it's seeing when it's dark and moving around, versus when it's lighter conditions, those same SOM5 cells don't inhibit the integration of, or rather the visual cortex, because it's light and you can incorporate the visual movement into your vision field as well, help differentiate between your head movement in the visual field versus something moving in the visual field. So that was only going to be.
He won?
I think he won. Well, it was kind of complimentary.
All right, everybody, that's episode number 29. Thank you for joining us, Guy. That was a lot of fun. We'll be back next week with another episode of So go ahead and tell us what. Yeah, go ahead.
Actually, it was actually good. Overall, it was almost perfect. Just necessarily not accurate in the terms of position. It's not position, it's velocity. So it's just the speed. I'm not looking at position. Even if you have an encoding in your brain of position, so basically you have some cells in your brain that will tell all the time in the outside world if you are looking like north, south, or wherever. In my case, it's more the velocity. So it's like you want to integrate both your head motion, so the speed, and the motion in the outside world, right? For instance, if you are, I don't know, a lion or a tiger, you have the prey, you want to accurately estimate the speed of the prey and the speed on your own motion to catch it, right? It's a sensory motor integration, basically. You need both. And so what you say is actually accurate. Somehow the velocity is also an integration of position. It's true. Because at the end of the day, when you integrate a velocity, you get a position. But it's not what I show in the paper. But overall, like the big picture was, it was it was good. It was good. So don't worry. He won, but he went into more details. That's it. So then you were talking, in your case, about the cellular level. So what is great with mice is that we have tools to manipulate neurons, circuit, and dissect circuits. And so you went to the cellular level, talking about somatostatin cells. So it's a very specific cell type that you have in a lot of places in the brain, but for instance, in cortex. So if you want also, I can define what is cortex. I'm not sure it's necessary, but at least specific part of your brain, in my case, that integrate visual information. And you have those cells that they do stuff, right? It has been shown for visual processing, but not much for other non-visual processing, because basically we start to discover since the last 5 to 10 years that we call that visual cortex, auditory cortex, somatosensory cortex, but basically they do many things. And we define them by their anatomy and the function, but actually you have a lot of multisensory processing. So the visual cortex can integrate auditory information, vestibular information. Vestibular is this, what I'm doing. Basically, it's what's your accelerometer that feels your motion. And it integrates all of that. And what I show in the paper is that this specific cell type can integrate both information, visual and vestibular. And what is interesting is that potentially this cell type could also integrate, for instance, auditory and visual. And basically, I wanted to see at the cellular level what was happening, and we found basically one cell type that was really explaining more or less the behavior. And as you said, one hypothesis, and we don't know, when you are in the dark, you don't want necessarily visual processing to happen. So you don't just silence your visual cortex. It's not useful. You're in the dark, right? You don't need it. And potentially, if you look at other area like auditory and so on, it would do the opposite, because you need in the dark your auditory system. Yeah. But then once you are in the light, well, maybe you want both. You want to optimize your perception, your visual experience. And basically your vestibular system will help the visual system to understand what it is.
So is there like an added efficiency that's gained by the brain by suppressing the certain, like when it's dark and you don't process the visual information? Does that add efficiencies to the brain so they can be better in other areas?
I didn't test it, so I cannot tell you.
Yeah.
What I can tell you is that even in humans, so it's kind of relevant, because even in humans, people describe that this suppression of the visual cortex during motion, and they call that the reciprocal inhibition. And basically what is happening is that there is no demonstration of that. In humans, it's very hard to do a demonstration of this circuit level. But what they claim is that it allows you to differentiate when you are moving from when the outside world is moving. And it's, if you think about it, I just, I don't know, take this. And while you are taking that, any motion trigger a sensation, any sensation trigger a motion, and it's a loop. Yeah. And at the end, you need to differentiate both. Because, for instance, in schizophrenia, you cannot differentiate both, and it's, it can start to have illusion. You don't know if this sensation is due to your own motion or motion in the outside world. And I think it's fundamental to have this motor system and vestibular system to talk to sensory system to differentiate the two. I don't know. Yeah. Did I address your question correctly?
Yeah. Yes? Yeah.
Guy, I want to thank you for fighting your instinct to move the camera. I saw you go for it, and you resisted and moved the water bottle instead. That's great radio instinct. I want to take a kind of a step back and talk more broadly about this notion of integration as it applies to neuroscience, and in particular, this way that the brain has to incorporate different signals in order to make some kind of decision. But also another book that Joe and I were reading this week is a book by a Dr. Bouvier called The Brain Inside Out, which is kind of explaining how our brain, rather than just taking in information, is at the same time, as you were talking about, it makes these predictions, and then it has an experience, and it compares that experience to the prediction. And I guess for me, what I'm trying to understand words like signal, words like integration as they happen in the brain, can you explain kind of at a cellular level what is happening to facilitate these things? How do neurons actually talk to one another, and how are different parts of the brain connected in terms of signaling?
Okay, so do you have 4 or 5 hours? No, so let's say that, as I was saying, at least in textbook, when you go to school, and even me when I was going to school, basically they define the brain as, and the human brain as the one is a cortical area that has been very pronounced, right? And that's why we are humans, because it's very developed, and you have a lot of neurons contacted to each other. And so, you also define the brain by different areas. And I think initially it's by the anatomy. So for instance, the visual cortex, you have the retina that sends projection to a relay zone that is called the thalamus. We call that a relay zone, but I think that's in a couple of years, maybe we should give them a different name. It's not necessarily a relay. A relay is like it's doing nothing. Well, it's doing stuff, obviously. Otherwise, you will not do that. You will go straight to the cortex. So basically, you have the retina going to the thalamus, this relay zone, and then the thalamus sends projection to the cortex. And the cortex, people say, well, you integrate for visual cortex at least different features. So for instance, if you have a bar that is horizontal, you will activate a certain population of cells. If it's vertical, you will activate another population of cells. And we define basically cortical area as with having in mind a really cellular level and cellular understanding, like a single cell does something very specific. And so, the thing is that when you record those neurons in cortex, you will have a small fraction when you present different stimuli that will. I take, by the way, visual cortex as an example because I think it's very easy to understand. But I could take auditory stimulation and it would be different frequency. I could take different smell for the olfactory bulb. It would be the same, more or less. But so you have the visual cortex with different features, and people started to realize, not recently, but as I was saying earlier, that those cortical areas basically respond to many other things, right? And it's funny because back in the days when they started to record, when you record the neurons, you stick an electrode in the brain and you will have a response of neurons. Basically, what you would get is only a fraction of neurons that will respond. And you don't know if it's because you don't provide the good stimulus or if it's because, I don't know, they respond to something else.
Or just measurement error, right?
Or measurement error.
When you're measuring on such small scale.
You can have ground truth more or less control for that. Depending on the technique, yes, you can have a measurement error. That's very true. And so, so basically, we define a different area by responses, but at the end of the day, we start to think that's one given area that will respond to visual stimuli, respond to actually a lot of stuff. So now, how do they communicate with each other? It was your question. So basically, between neurons, you have synapses. So synapses is just a contact with neurons, and you have a different type of synapses, but I will talk about the most famous one. And you will have some molecules with the electrical signals that would be released to communicate with the other neurons. And you have a presynaptic site, so pre-synaptic site to the postsynaptic site. And then here you can have also basically communication. Depending on how much you stimulate, you can have different type of synaptic connection and synaptic plasticity too, because obviously our brain is plastic. And our brain, like a neural network, can learn, right? And depending on the data that you give to the brain, then the brain will make predictions. And I define the cortex initially as something very passive. You give a stimulus, you have a response. Well, it's actually more and more people start to think that it's not like that at all. You make prediction all the time, and visual prediction, for instance. I don't know. I'm in the hallway. Every day I walk down the hallway and I see this painting on my right. I'm pretty sure that if in one day I remove this painting, you will really pay attention to it. Otherwise, you won't even see the painting.
Mm.
And basically, you make prediction all the time. If I'm walking straight, I expect a visual flow going this way because it has been like that all my life, right? I don't know if I turn the head and something is vertical, I expect something to stay vertical. If it moves with me, I would be scared. I would freak out, right? Oh, what's happening? So basically, you make prediction all the time, and your brain is very good at making prediction, and you have actually some structure, but it's not very necessarily very well understood yet. That once one can give this, there are prediction machine that would give this prediction signal to the cortex, for instance. Or some people say that it's happening in cortex, some people say it's happening somewhere else. I think it's happening everywhere anyway. But basically, your brain is a prediction machine. It's not just you are not a snake, right? It's not purely passive, right?
It's not just responding to the environment around you. It's creating that environment.
Yeah, and as I said, why it's important to what I think what I'm doing is important is because you want to, because of your motion, your own motion, you want to make prediction about your sensory expectation. Mm. And both of them are really tight together. So I think we start to really think, we used to think at least 10, 20 years ago as this different brain area, isolated, you look at the single cell level. Now we look at the population level using different type of sensory signals. It could be visual, auditory, motor signals, and try to make sense of that, like really at the population level. So it's a very exciting time, I feel, to be a neuroscientist. And even if I started my PhD on something very cellular, because I wanted to understand how one single cell can integrate this different information, because even one single cell can have auditory and visual, for instance. But now I would like to make sense of that, like really at the population level, because I think that's how you will understand maybe one day how the brain works.
Well, and Guy, just one example, I think, for our listeners of what it means for your brain to make a prediction and then for that prediction to be compared to your environment. Yeah. Everybody's had the experience of picking up what they thought was a full cup of water and just, almost throwing it because the cup of water was actually empty. We were expecting it to be heavier. We were prepared. We made that prediction, and then we almost throw it off into orbit or something. So this is an idea that all of us are living with, where we're constantly making predictions that are either validated or invalidated by our environment. And then we respond accordingly and make another prediction, so on and so forth.
Yeah, it's like, it's think about tennis. If you play tennis, you will correct your movement because it was wrong, because the ball went outside, right? And then you basically make prediction every time about your movement and have an expectation about what would be basically the trajectory of the ball. And you optimize basically your prediction all the time. The thing is that you are not exactly always in the same position. And so that's why sometimes you make mistakes. If you were exactly in the same position every single time, you will recruit the same exact circuits. And ideally, it would be a win every single time. But obviously, it's not the case. But yeah, that's more or less the idea. And it's not necessarily, you have also animals with less cortex, like humans, or less developed cortex that are great at that. And so it's also telling us that you don't need necessarily to be a human or cortex to do that. And mice is, for instance, a very good model to study that.
You were talking about the visual processing and how the mind or how the brain pieces in certain pieces of information that might be missing, or it makes predictive models of what you're seeing.
Yeah.
So does that mean that just because I see this field of vision in front of me, like how much of that is real versus how much of it is maybe abstract or made up or missing? You understand what I'm asking?
I see. At the end of the day, it's
like what's real?
Perception is not real, right? It's biased by your experience, what you've been through, your movements. So for instance, if you want, some animals, they have very bad binocular vision. So meaning that both eyes communicate with each other to see things in 3D, for instance. And you can see those animals that will actually start to move like that.
Size you up.
Exactly, to basically see how far it is, where it is in 3D and so on. So basically, also your representation of the world will will be affected by your motion. But of course, by the context, by the experience, I would see different stuff in different contexts, right? It's you have this. I have a friend that plays a lot. Oh, what's the name? Squash. And every time he plays squash, basically he put glasses, to protect himself. And so once he forgot his glasses to play squash, and he was telling me that basically everything was a bit shifted, and it's because when he plays squash, he has glasses. He's used to it, and
You
so
have you have that parallax error coming into your
exactly
glasses.
exactly.
Yeah.
So it's everything works with context, but thanks we are quite plastic, especially with eye movements. In a couple of minutes, it goes back. It's like if you go to space, like yesterday they sent other astronauts to space. When they will come back, yes, and the head of the new mission is a French.
Oh, there you go. Perfect. Hey, perfect.
Yes, and so when they come back to Earth in a couple of days, initially they feel really bad gravity, right? And in a couple of days, it's back. It's back. And it's another example, by the way, of how vestibular system, because vestibular system helps you to feel also gravity, can affect your vision. They can have real trouble in seeing stuff when they come back, or when they go there as well at the beginning. But then the brain adapts because it's an amazing machine. Right?
Well, one of the things that is interesting to me is to understand it, at least from kind of the outside looking in, it seems like there's been a lot of kind of excitement around neuroscience the past decade or so. Maybe that's not true, but that's kind of how it seems to me kind of on the outside. And we talked to another one of your lab colleagues about some of the breakthroughs that made their research possible. And I kind of wanted to ask you the same question. What key technologies allow you to do the research that you currently do? And another question tied to that one is what technology do you think right now is lacking that if it were improved would make your research better or easier to do?
Yeah, so I think it's, as I said just before, I think it's a very exciting time to be a neuroscientist. And it goes fast. It goes really fast. So even when I started my postdoc, I was using this kind of electrode to record the brain activity, and you have small channels that are very close to each other, like 20 micrometer away to each other. So very small, and these electrodes are very thin, right? And so the maximum that we could use, and that was commercially available, it was 32 channels. That was like, wow, a lot. The postdoc just before me, they were using 16 channels. And for them, it was a lot. And so I was using 32. And today, people use thousands of channels. So it goes in 5 years. I started my postdoc 5 years ago. So it goes so fast. And so I think that I haven't done any real breakthrough in terms of technology for my research. I think I potentially did breakthrough in terms of scientific results. But I think what was new, or at least you don't have a lot of people that are doing that in the world, is putting together specific things like to stimulate the vestibular system to trigger motion. It's a passive motion. And to do so, I have a servo motor that controls a turntable, and I have an animal on the turntable. It's like, it's like playing games. I mean, I could even put a wheel, and the animal could drive the turntable. And around that, I have multiple monitors to stimulate the visual system. And at the same time, I do recording in multiple places in the brain. So I think putting together all this technology together, basically putting that together, was the novelty. It was the novelty in my lab. I was the first to do that. And I think in the world, in mice, you have only a couple of people doing that. There is one lab in London that works. I mean, they started that a while ago. They are very good at it. It's the lab of Troy Magri in UCL. But otherwise, you don't have many people. So it was initially, at least for me, quite challenging. And I think that we are going into the right direction. So what could help in the future my research, like technology? Well, here it was a passive movement. right? I'm controlling the motion. It's very good because you want to very accurately manipulate the parameter space. And you have one parameter at a time, meaning horizontal movements, one direction, one speed. If you have an animal in 3D moving, it's going in all directions, and you don't know if horizontal axes integrate differently when the vertical axis is used at the same time, and basically how in 3D it interacts together. So it's way harder to understand, to make sense of it. And we can do that. Actually, in my paper, we also did that in the last piece of data. We have basically a chronic implanted electrode. So you implant the electrode and then you leave the animal. And at the same time, I put a small accelerometer on the head, so I can record head motion at the same time. And then you can try to see a correlation between movements and activity in the brain. So that's great. But at the same time, when I put that, the weight of this stuff is 2 to 3 grams. For us, it's nothing. For an animal, it's
on your head or something.
And on top of that, you have a cable.
Right.
The animal, you will notice something. Basically, if you do, you look at an animal that is randomly like behaving, and an animal that has something like this on the head, they will do way less movement like that.
Right, right.
Obviously, they will do it because they are used to have this cable and so on, but way less. So it's still not perfect. And I think what would be amazing is to have a wireless electrodes with tons of channels, like really tons of channels, like to record as many neurons as you can, with this accelerometer that you have on the head, on the arm, everywhere on the body, basically.
Yeah.
Or having many cameras, and people are doing to do that, are starting to do that. And then you can just reconstruct the brain in 3D. The issue with cameras is that you need really high-speed cameras, and many of them. So, but I'm sure, I mean, we can send obviously people to the moon. So we should be able to do that at some point the next years.
Very good job. I'll pass it back to you.
Yeah, so I would like to take a little bit of a step back and talk more about the academic process in itself. And you've been in several labs and several groups, and I imagine you've had several advisors or PIs, I think is the proper term. I'd be curious if you could talk through us like what you look for as you select a PI or an advisor or determine a lab that you're essentially going to spend the next few years of your life working with. Like, what is it about the good PIs that stand out to you? And
so
and
how
yeah,
do I pick a lab, basically?
Yeah, how do you pick a lab, and then also apply that to like how do you pick a project like this one? Like how did you pick this particular endeavor?
So basically, I started to be so I did a lot of competition in table tennis when I was younger, and I always been fascinated by sensory motor integration, and especially sensory motor learning. How do you optimize? I was talking about tennis because people I mean, can clearly understand tennis. I mean, table tennis would be exactly the same. So how do you optimize the movements, right? And so that's how I got really interested by that. And that's, I think I wanted to understand how it works. And the first rotation that I have done actually was not necessarily about this. The first rotation that I've done, I was like, oh, I want to do research. Please give me a shot in your lab. So I was not necessarily very passionate about the subject. And actually, after a couple of months, I asked the PI, oh, do you know anyone doing neuroscience? Because I don't necessarily, I didn't say I don't like what you are doing, but I don't necessarily, I think it's not what excites me the most. And unfortunately for me, her husband was a very famous neuroscientist. So I talked to her husband. And so I did my first big internship was in Cambridge in UK. And I was working on something actually kind of different again, but I'm getting there, no worries. I was working on multiple sclerosis. So this disease that basically you have
MS.
MS. Okay. Okay. Yeah.
So you have a degenerescence of your myelin around your neurons, that will basically avoid any transmission between neurons, and at some point you die. And we actually don't have a good, very good treatment for that, as most of the neurodegenerative disease, by the way. And so I felt that, at least initially, I always felt that I need to have something to work on something meaningful and useful for people and could help people. And so I started my first rotation, let's call it an internship, doing that. But then I was like, I want to record neurons. I want to see activity with my own eyes. I want to, see basically the neurons being active. And then from that, I really started about, like, this is really what I want to do. And then I had a very different approach to picking a lab and picking a PI. And here it was really about, and it was more important than an internship, because then it was my PhD. PhD is long, so you need to be right. I'm not, yes. And so I needed to be fascinated by the subject, obviously, but also being very inspired by the boss, by the PI. I think it's the first choice. Then the second step is you start to talk to people in the lab, and you want to know, is this guy an asshole or not? Basically, that's, yeah, but PhD is long, so you want to know. It's not necessarily obvious from outside. He can be a very good scientist and basically being really an asshole. So you don't want that, obviously. And then you start also to ask people, basically, is it a good environment? Do you have like people, are they helpful? Are they inspiring? And all this kind of question. And then, oh, well, do you like the city? It's important too, right? Can you have fun in this city? And if you have a +1, well, you also want to discuss to a +1, obviously. And
not to cut you off, but you're saying that you want the PI to be inspirational and like the group to be inspirational. Can you break that down? Like what is it in the PI that inspires you?
So I would say my first, for my PhD, for instance, I was fascinated by sensory motor integration and learning, and I wanted to understand it at the cellular level. So I started to work on synaptic plasticity. It's this process where you change the strength of the connection between neurons, basically to optimize the motor command, to have the perfect movement to play tennis, for instance. And you will adapt and optimize the synaptic weight between the neurons to have something perfect. But I wanted to understand the cellular and the molecular level. And my PhD advisor, I had 2 PhD advisors, but they were both my teachers. And I found their lectures fascinating. I was also very impressed. I found they were very charismatic. And it's not necessary, but I don't know, to me, at least, I'm sure it made a huge difference too. But I think for my PhD project, I was just fascinated by the project. And I really liked my PhD advisor. I thought that they were inspiring in terms of question, in terms of knowledge. They were very knowledgeable. And as a human being, they were very good people, a very good person. So I talk also about, as are they assholes, are they good people, and so on. For me, it's something that is very important. So I cannot work with someone that I don't really like. So some people can, I cannot.
Well, especially for a PhD, it's going to be 4 years, right? So you want to be, yeah, exactly right. Joe, I know that we have an album of the week, and I think kind of the next thing that we want to get into with Guy is talking more about this research area. And this is going to be kind of a bigger topic. Why don't we give our album of the week real quick, and then we'll hop back into this conversation. We have a special album of the week in honor of our guest, who is a very charming Frenchman. And to honor that, we have an album of the week, Joe, that I think he will find very enjoyable.
Yeah, so I'm excited to see if you've heard of this album or this band. So the album is Tiamo, and the band is Phoenix.
Phoenix, yes.
Okay, that's a good start.
Wait, Joe, just stop right now before we ruin it.
Okay, so for the notes for this, it's synthy, Italian vacation, lots of gelato references, groovy and dancey, romantic, cinematic lyrics, 80s soft rock. 80s soft rock lyrics are also slightly darker than the sound, upbeat but not rapid pace, unrequited love, and then sultry. And then jumping into the review, with the current state of the world, we could all use a vacation right now. This week's album, Tiamo by Phoenix, is loosely based around a trip to Italy. Tiamo is the sixth album by Phoenix and was released in 2017. Inspired by Italian disco and soft rock, Tiamo tells the story of an unrequited love. The first two tracks on the album, titled J-Boy and Tiamo, set the stage for a whirlwind romance. They are both upbeat, fun, sultry, and irresistible. You know the feelings are unsustainable and probably unhealthy. However, lust does tend to have a blinding effect. On the track For di Latte, Phoenix relates the decadence of making love to the silkiest Italian sweet cream gelato. Maybe Costanza was onto something when he tried to mix food with pleasure. By the time you read the track San Soleo, loneliness begins to creep in. With the final track, Telefono, the harsh realization that the romance is over finally hits. While the lyrics describe a one-sided relationship, sonically, Tiamo is a really fun listen. The bass is punchy and creates an up-tempo groove. Pair the bass with the congas on the tracks such as Tiamo, and you can't help but let the rhythm move you. Then there's the synthesizer that mixes in an airy and effortless sound throughout the album. Overall, Tiamo combines the effervescent feeling of infatuation with the darker reality that it might not last all while you're dancing the night away in an Italian discotheque somewhere in the 1980s. For a solid listening experience, I'd recommend grabbing a pint of ice cream and dreaming of your next big trip once everything settles down. So, and just to remind you, we don't write this. We have a correspondent that handpicks these albums for us each week. What's your take? Are you familiar with Phoenix? And
yeah, a bit, but I didn't even listen to the album, so I cannot really say anything. So unfortunately, I just know Phoenix. I listened a couple of songs like a while ago. Yeah, now that I have a baby, actually, I don't listen to much music anymore. That's bad. Not surprised, and then you wake up. He wakes up. So.
Fair enough. So Joe, I don't know if you had a chance this week to listen to the album. I had a chance to listen to it, and of course we'll put all this information below the video. I really enjoyed it. I thought it was a fun listen. What was funny is, as you were saying in the lyrics are in Italian. And I knew that it was a French band, and I thought I was like having some kind of weird stroke or something. I'm like, I'm hearing Italian, but it's supposed to be French, and I was really freaking out for a few minutes. But
there's also like English lyrics in it too.
Yeah, I thought I was having some kind of flashback or something. It was really a lot of fun. No, it was a very fun album to listen to. And Joe, another album to add to your future coming day of the all album experience that you're putting together. So this would be good to have during dessert, I guess. During a lot.
Any accent when you speak English, for instance?
When they speak English?
I feel like I didn't really hear much of an accent. I don't. Maybe
yeah, I feel like every time people sing, their accents go away.
Yeah,
like a lot of these British people.
Yeah, exactly. It's what I feel too. And every time I'm impressed. And then, I think I heard interviews and they speak kind of nice English. I mean, I cannot really tell in contrast with you, and it's easier to pick any small, detail and difference. But yeah, I feel that they have a pretty good, which is quite unusual for French to have like very tiny accent.
Yeah, it was a lot of fun. Definitely a good album, and of course we'll put all the information in our tweets and everything else going out. But to get back to where we were kind of talking about before, talking about kind of the research environment in general. You were talking about what you look for in a good PI, and let me kind of follow up with that question about what you look for in good lab mates and in good fellow researchers. And I guess to kind of set the table, my question would be, what is a good size for a lab group in order to facilitate cooperation between the different researchers that are involved? and how important is that cooperation among different lab members to bring about the best results for the different areas of research inside of the group?
That's I don't know. That's a very good question. Um, so what do I expect first from I mean, colleagues? Well, being inspiring as well, right?
Yeah.
Learning stuff from them. It's even better. And I think my postdoc advisor, so Massimo Scongiani in UCSF, he's actually picking people this way. He's picking people that are doing not necessarily the same thing.
Yeah.
And try to put them together, and he will learn from them, and we will all learn from each other. And I think he's doing a really good job. And if you look at his publication track, obviously he's doing well. He's doing a good job. The way it works in my lab here is not necessarily the way it works in every lab, meaning that we have a project and we don't do much collaboration inside the lab. We have different projects and we try to not necessarily overlap too much between lab members. I think it's good, and at the same time, it also avoids conflicts, because at some point after you're a postdoc, you leave the lab with a subject. And if you have two people working on the same subject, well, then it's the octagon, right? And then it's and then a fight, right? No, it's I think it's good, at least the way it works in this lab. I think it's super good, but I think it depends also on the question. You can have some projects that are very demanding in terms of basically, in terms of work and quantity of work, and you need to team up. I think UCSF is quite collaborative. I've done already collaboration with other labs like Vikas Soal in UCSF. I'm doing collaboration right now with Kevin Bender working in ASD, so autism disorder syndrome, and the involvement of the cerebellum, brain structure that is involved in motor learning. What is the involvement of some synapses in this behavior for autistic kids? At the end, it's try to make sense of what does this mutation in these kids. So I think it's very collaborative in terms of teams, not necessarily in terms of people inside the lab, but it's not always the same. In my lab, at least, it's not working this way.
Okay.
I'm not sure it's the best, by the way. What I'm saying is that it works quite well here.
Sure.
But I don't know if I would necessarily do that in my lab, for instance.
Sure.
So that's a good segue to my next question, which is you're getting to the point in your career where you're looking at starting your own lab, correct?
Yeah.
So when you start your own lab, what are some things that you, especially the considerations that you want to take when running the lab? Like, are there certain tenets or rules or certain influences or anything like that that you want to maintain in that lab?
I don't know. I think having being really open with people in terms of your project and having continuous scientific discussion, or any discussion actually, between lab members is important. Some people, they have a tendency to not really talk about their science because they are afraid that these people will talk to another scientist, that will talk to another scientist. At some point, someone randomly in the, working on more or less the same project, somewhere else in the world will hear about what he's doing and about the result, and it's not published, and you don't want someone to scoop you, basically. But if you have, like, at least inside the lab, I want a continuous brainstorming between lab members. I want it to be very open. As a PI, I think that's one of the first things that you should keep in mind, is to really try to be fair with people. You can be tough with people, that's fine. You can ask a lot, but be fair with people. I think that's the first rule, because something unfair is really upsetting, right? And so I think I would really try to be fair, with people, and try to help as much as I can. But it's also different when you start a lab and when you have a lab since 10 years. When you start a lab, you need to be really hands-on. You need to push, because you want to get the tenure, right? You want to have your job. You want to have a secure job. So I think you need to really push at the beginning. So I don't know, I will most likely behave differently at the beginning and after 5 or 10 years. But I don't know, otherwise, I don't know, do good science. Like what you do and being excited about what you do. I don't know. There is no real, it's like starting a company somehow. It's very exciting.
Well, that's a really good segue because I do want to make kind of the comparison between a research lab and a type of startup, which I think there's a fair comparison there. But before we get there, I would be remiss to not kind of hone in on a thing you were just talking about, which is this notion of competition. Um, One thing Joe and I talk about a lot on the show is kind of the pros and cons, if you will, of competition. And two writers that I find very influential, the first being Peter Thiel, and the second being a French philosopher by the name of Rene Girard, talk about this cycle of competition and kind of in extreme cases leads to violence. And more or less, it's this idea that competition can cause kind of an obsession between groups of people that were otherwise on kind of separate tracks, but through competition end up focusing on the same thing. And this idea of being scooped kind of reminds me of that in the sense that you may have two researchers that are in similar fields, and yet they hear from somebody else and they think, oh, that might be some big breakthrough. Let me kind of change my research to do what they're doing to maybe beat them to it. And the next thing you know, you have two researchers that were otherwise doing important work are now focusing on the same thing. And I was just kind of curious to get your take on that. What is the role of competition in academia right now? And do you think there's too much competition? And do you wish that there were more collaboration between different groups that were researching similar topics?
I think it's not even about what I wish, but what can be the most efficient.
Right, right.
And I think that competition is obviously a good thing in certain contexts. In economical context, I would say it's a disaster. I don't know. I mean, we are not here to talk about politics, but somehow we could here. But I won't talk about it. But in any case, competition could be good because basically you want to go as fast as you can. You want to push. But at the same time, in science, doing so, you have a lot of people that are not necessarily following the rules to get the result that they want. Why is that? Because you have a massive pressure on competition to get funded.
Sure.
And then it's a loop. You need to publish very well, a lot, high impact journal to get funded and to get the PI position, and then to be able to have money for your labs, and then to have money for your labs members. And then it's circular. And at the end, I think that we put so much competition that you want to go where basically you will have a result really soon. Because even the next year you don't have funding, you're dead. So that's how you want to explore basically, and that's how you basically limit the discoveries. You would make way more discoveries potentially. I mean, not having that many competition, that much competition.
So what does it look like for your research to get funded? Like, what are the parameters that are being evaluated on whether it gets money or doesn't get money?
Very good question.
Or is it arbitrary?
No, it's not. Obviously, it's not. Sometimes you think it could be, honestly, but I don't think it is. It's arbitrary in a way that the people evaluating you are specialists that are not always specialists. But if you write very well your grant application, well, anyone should be able to understand. So try to write it very simply, very well. The issue is that it's biased anyway, because you will have two, maybe three people that will be evaluating you, and maybe they are specialists. So meaning if they are specialists, they are kind of a bit in competition with you. And even if they are not in competition with you, it's biased because you have two or three people. And even if you have only one people that doesn't like it, so for instance, when you try to publish it in very high impact journal, only one person doesn't like your manuscript, your paper won't be accepted.
Does that like prevent you from taking risks just because you don't want to offend that one squeaky wheel out there that might?
Oh yeah. I mean, taking a risk, I think, you have two ways of doing science anyway. You have a result, you analyze your results, you make a prediction about the very small, tiny next step after this result. Okay, how does it work? And then, and so on and so on. Or you can have like, okay, I want to understand how it works. We have no clue about anything. And I will just start to record. It's more or less, well, it's not more or less what I did, but somehow it's a bit related to what I did in actually my paper. We don't know really what it's used for. We know a bit the circuits, but we don't know what it's used for. And I think if you bring what it's used for, that's how you get to the high impact journal. It's not necessarily super easy to get there, especially when you have something very exploratory.
Right.
But for sure, I mean, doing exploration, for instance, RNA, studying RNA. And so today with COVID-19 and the vaccine, we start to discover that, oh yeah, that's important. But the person that actually did a lot of work on that, I don't remember her name. She was in academia. I think she left after that. But anyway, basically she didn't get funding back in the days.
Right.
And so they were like, oh, it's not interesting. It will never work. Well, no, it's more or less saving the planet. So, you know, it's very arbitrary. It's what I'm saying. I don't remember her name. I'm sorry. Exactly what was the different steps, how it works. But basically, this is more or less the spirit.
Right.
What we are doing. Some people will say it's not useful, and we won't get funding. Even scientists will say it's not useful. But maybe in 10, 20 years, 30 years, I don't know. It might be useful for something. But that's how science should work. But the thing is that money drives the world. So what can you do?
And does most of the funding, does it come from government type agencies or state agencies?
Not necessarily. It depends on the country as well. In the US, you have, for instance, this building where we are, you have a lot of private fundings. And so you have a lot of private fundings in the US. And I think that countries like France should try to go into the same direction because the public funding is very, very low. And you have a massive competition to get, like, on top of that, not even a lot of money. So,
Right.
So yeah, it's you have both. I think that's here. It's rich people that are giving money to make this kind of building, for instance. But what would be great, and I think it was kind of related to maybe your next question that you already just started to talk about, private company and basically academia.
Yeah.
I think we should go into this direction because they know how to make money. We don't, obviously, but I think we can work together on it could push really further the science.
Well, that's and I think that was kind of, let me just ask one more kind of general question about research, and then I do want to talk a little bit about thinking of research as kind of a company. But what would be your pitch to the public about why this kind of research is important? I know we were talking with one of your lab mates a few weeks ago, and the topic was brought up about the importance of communicating research to the public in kind of in a way to show why it's important, but also to try to demystify some of it as well, which I think is also important. What would kind of be your elevator pitch or something like that, to the public to explain the importance of your work and really what motivates you as a scientist to pursue this kind of work?
Yeah. So if I take the big picture of sensory motor integration, I will tell you that most of the neurodegenerative disease, Parkinson's disease, autism disorder, schizophrenia, you impact your sensory perception of the world, and it's also sometimes related about how you integrate basically your vestibular motor system and your sensation, so it could be visual or anything around. So I think what I'm doing right now, I'm not using as a model system any gene that are involved in any of these diseases. But if you want to understand something that is not working, you want to understand how it's working first. But I think people want results in the next 4 or 5 years. It's like a politics. He will give money to education and money to research. I mean, actually they won't, because in 5 years they're not there anymore. They won't get the reward. They won't be congratulated. Oh yeah, you put money into research. It's good for your country. It's very good. No, it will be in 20 years. Well, they don't care about 20 years. It's not about that. It's maybe a Republican or the other one that that will basically have the results. So basically, it's hard for people to understand when you don't have an immediate like right now a result because it's how it works in private company. You have a deadline, you need to have a product, sell the product, and the product has to be useful to make people happy or to facilitate life for people. So I think that's why. Or otherwise, you are very, very, very rich and you have a lot of money on your bank account, and then basically money will bring more money. And you do whatever you want with this money and you do research, but it doesn't work like that.
Very good.
Yeah, no, I can see that as being a
Yeah.
tough business model, like especially for disciplines of neuroscience that cater towards some of these neurological disorders like MS or schizophrenia,
Yeah,
like you mentioned. And it's like people, like investors, aren't going to want to put money towards that until they become affected by something like that, until
exactly.
they get some skin in the game.
Exactly.
And then at that point they care. But other than that,
I think you have the CEO of Red Bull, for instance, is I think a family member had a car accident. I think I'm not necessarily sure, but he had an accident, and then he had like basically this what is impacted in the myelin around the neurons. Basically, it has been cut. The spinal cord has been cut, and he started to put tons of money in MS and in basically stem cell research. Basically, the idea would be to inject stem cells to create new myelin around the neurons, and then basically your spinal cord that was cut like that will start to regenerate, and then you will walk again, for instance. And so yeah, usually you have very, very rich people, and they have a family member that got cancer, that's MS or an accident or whatever, and they start to put money. I think it could be maybe, but as I was saying, in the US, honestly, a lot of money from very rich people is, it's easier to give money actually for research in the US compared to France, I feel. But
what type of roadblocks are there? Are there like taxes or?
Well, taxes kind of, yes, and maybe how to say when you don't want to.
Intimidation
I don't know why exactly in France. I don't know. It's not how it works. It's not like in the 16th century where you have like these rich people that were giving money for I mean scientists. The light century basically. I think it should work way more this way, because obviously the economical how it works right now with public funding, and it doesn't necessarily work. It's what I'm saying, because of the business model basically. Or otherwise we change the business model, and private and academia basically can work a bit more together. But I think for the long run, we need potentially change or optimize the business model of academia. If we can call that a business model, by the way, we cannot.
Well, so I guess kind of tie in, or to kind of tie in this segment before we get on to kind of the more personal impacts that your research has had on you. Let me just kind of ask you kind of one more tying up question for this, as a result, or as it relates to the role of innovation in research groups and in being a good researcher. And you talked a little bit about this earlier, about there not being really a key breakthrough that made your research possible, but it was more your ability to assemble a collection of technologies in order to arrive at some kind of novelty. And that reminds me kind of this theme that I see emerging right now, that a lot of the breakthroughs, especially in biology, are not necessarily going to come from peering deeper into a problem, but in understanding how different systems work together. And as we were talking about the brain earlier, that seems to me a very natural place for this kind of thought process to be very important. Rather than understanding more and more about how an individual neuron works, it'll be more and more about understanding how the brain works at a systems level inside of the brain. Do you think that's a fair assessment in terms of innovation on the one hand, and also the systems perspective of the brain, or do you not agree with that?
So I think there are more or less two questions here. So the first one is, I think it's important to understand the technique and then put them together. An engineer will not just, but will basically make the technique and make things happen, right? But if it was the case, and if it was what we are looking for in neuroscience, we would just hire engineers, which could be, by the way, an idea that is not stupid at all. As for instance, for Neuralink, the Elon Musk company, you have a couple of neuroscientists, but you have mostly engineers.
Okay.
because the neuroscientist will think about the question, he knows the brain, he knows the circuits, he knows how to do experiments in animals, but the engineer will just build the electrodes once. I mean, just again, sorry guys, but we'll build it's mandatory, right? But we'll build the electrodes. So we need both working end to end, basically, and that's how you make discovery. And I don't remember if it's Sidney Brenner, the Nobel Prize for apoptosis, that say the sentence like basically you made discovery because of breakthrough in technology or something. It was not the sentence at all, but it was something like that. And I think a lot of scientists think this way, but I also think that these days you can have people, oh yeah, I can record 1 million neurons at the same time. Okay, good for you. Then what? What is your question? What do you do with that?
Yeah, where's the application?
What is the application? So you can have many, I mean, technologically, and that's very good because someone else will say, oh yeah, now we can do that. And someone else thought about, oh, once we have this technique, we can use it. And that's how basically someone else will think about using this technique for his question, for a specific question in disease or something else. And I think that's why collaboration are important. And but it's not alone that you will have a breakthrough and then you will have an idea. It doesn't necessarily work like that. Someone else will have a breakthrough, and you need to have a big picture about what people are doing around to pick what is interesting for your question. And I think that's how you push forward the science. And then the second question, what you were making an analogy between, once again, the brain, right? Different areas. That's
Right. Just kind of if what you thought kind of the future of neuroscience would look like, and do you think it would be more and more about understanding how different parts of the brain work together, or okay?
Yeah. So basically, I would say that in the 90s, it was really about I will record one neurons and I will understand how this neuron integrate different inputs and what receptor of molecule and so on do what at the single neuron level. And it was because, and thanks to the breakthrough with the patch clamp recording, once talking about technique, Nobel Prize in 1991 about patch clamp, Sackman and Mayer in Germany. Basically, they discovered a way to record neurons and currents, small currents inside the neuron. And because of that, then you have like years of beautiful discoveries thanks to the technique. So you see technique and brain discoveries. And then we started to record more and more neurons in one given structure, but it was still in the intact animal. You record neurons and responding to one orientation, for instance, of the visual system, for instance. One cell respond to horizontal bar or another cell vertical bar, but it was still this single cell characterization. Another Nobel Prize for that was Hubel and Wiesel. It was actually way before the patch clamp. It was 1981, I believe. And but it was still single cell, right? And you have this characterization. And in the past years, it started to be one given area of the brain. We understand at the population level what is happening in this given area. And now it's like, okay, how areas communicate with each other, and basically the loop between different areas in the brain. And I think we are really going into this direction, which is, by the way, way more complex to untangle. And you can tell also with the duration of a postdoc, for instance. Back in the days, a postdoc, it was 2-3 years. Today, it's 5-8 years. It's because, I mean, the questions are way more demanding.
Right.
And you record multiple areas in the brain, and people want to understand from the cellular level to the most like integrated behavior, and it starts to be complex. But I mean, brain is probably quite complex, so questions are getting complex.
Yeah. So we have one more segment we want to get to, which is kind of going to be to kind of tie in Guy's research with kind of his personal outlook on things. And before getting there, let's do our social media shout outs real quick. And I think we'll both agree that the two behind the scenes stars of today's show, without a doubt, would be Francisco Aparicio and Cameron Wilhite, who were instrumental in making this episode come together. They helped set it up. They're both research colleagues of Guy. And so we'll be shouting them out on our tweets later on. But a huge thanks to them for helping put this together. This has been a lot of fun. And Joe, you were actually out there, getting this all set up. Just tell us a little bit about what it takes to make a show like this come together on site.
Yeah, well, there's always going to be unknowns when you go somewhere new to record something like this. But fortunately for me, I had producer Cam with me. I had producer Francisco, and we were able to find an awesome venue to do this, and an awesome interviewee to have on the show. And yeah, these things just have a way of working themselves out.
And here at R&R, we are always about the cat landing on its feet. And we've been landing on our feet now for 29 episodes in a row. Going to keep on going strong. So let's break into this last segment, and then we'll leave you guys to enjoy the beautiful, although slightly overcast skies of San Francisco. So Guy, where I wanted to take this next part is just to kind of get an idea of how your research has affected your perspective on things. And I wanted to kind of open up the question with asking you about how you think about the brain in terms of kind of like a mental model. How do you approach solving problems in the brain? How do you organize your thoughts around the brain? What kind of analogies or metaphors do you find useful for working through these kind of problems?
So I'm not sure. So what do you mean exactly by analogy? So you mean like the process, like well, it's more or less a scientific process.
Yeah, exactly. So kind of just when you're trying to pose a problem about the brain, how do you kind of organize your thoughts around it? I mean, obviously you were just saying a moment ago how complex the brain is. What kind of simplifications or what kind of generalizations do you use for trying to parse your thoughts about the brain, just to kind of help organize your thoughts around these complex questions?
Well, I think that's where you have to be careful, and being into more complex system is a lot of people, they have very strong conclusion when they see a correlation.
Yeah.
So for instance, I don't know, it won't necessarily cover all your question, what I'm saying right now, but I think it's important to say, and a lot of people say, well, if the animal is doing, I don't know, is grasping something, and at the same time, you will have an increase of activity somewhere in the brain. I'm sure you will have somewhere in the brain increase of activity. So you will say, well, it's because he's moving the arm. Well, at the same time, you move the arm, you move the muscle in the neck, you move the tail, or whatever. And so it's something that is very difficult. And that's why I think, well, before making conclusion, you should always try, and I think it's what we try to do in our lab. So basically we try to really see the correlation. Then if you have, for instance, neuron A or neuron B, you try to move neuron A, either inhibiting neuron A or exciting neuron B after the correlation, and then trying to make sense of this correlation, basically. And at the end of the day, what is important as well is, what does it mean? You have, okay, I have a correlation between these neurons and this behavior. Okay, then what? Good for you. I mean, it doesn't mean anything. So I think, and it's again something that is in the air as well. People are pushing the technology. I can record 1,000 neurons, and then I can decode the activity of the brain. And because I have so many units, I can decode it at the very, very good accuracy, like almost perfect. Okay, good for you. And what is your scientific question? What is your question? What do you want to solve? What problem do you want to solve? What does it mean exactly? So I think that's what I try to keep in mind when I ask a question.
So in other words, you're trying to avoid being just a basic cartographer or someone that's just mapping a region.
At least it's what excites me. I'm not saying that people are doing, for instance, descriptive description of anatomy or description of the circuits. I think that's very useful for everyone, and especially for my job day to day. But it's not necessarily how I see science. It's not necessarily how I get excited. Some people will get like, seeing a new circuit on a pathway could blow their mind. Well, me a bit less, and I'm more interested about the scientific question. What does it mean exactly?
Okay, so going back to worldview, I have a related question to that. You were talking about table tennis earlier and
Yep.
maybe tennis sports.
Yep.
When you play these sports, or when you do these tasks, these events, do you take anything you've learned from your neurological studies on sensory inputs to apply it to your game? Like, for instance, would it make sense to wear earplugs when you play so that you can be more focused on the visual part of something, or play blindfolded, or I don't know,
Okay.
do you take those considerations into activities like that?
Well, I played table tennis a lot before I did any neuroscience. I
Yeah.
think it's the opposite way. It was because I was playing that I got interested by how it works. Exactly. I think putting earplugs or any covering any sensory modality while you are playing table tennis or tennis would be a very bad idea, because you use actually your hearing to get an idea of the trajectory, especially in table tennis, because it makes a lot of noise. And the timing between, you hit the ball, you can hear the ball, you can see the ball, you can actually you use your audition. And I'm pretty sure it would be actually a fun experience. We should wear earplugs or not, and then play table tennis. I'm sure it would be terrible.
But Guy, how would you train a mouse to play table tennis? There's no way. I'm just kidding.
I'm working on that, but I'm sure maybe at least with monkeys, you can put electrodes in the brain, and then you can play. What's the game?
Pong.
Pong. Yes.
I'm sure they can play table tennis with a fake player, just thinking about table tennis. Because at the end of the day, pong and ping pong is very close, right?
Yeah.
It's only one word different.
Only one word, only one syllable. I mean, how? Yeah.
And so I think you could. I think one of the goals initially when I started my actual project was I want my animal to drive the table in the virtual reality. Because when you think also about my project is about visual and vestibular integration, well, what happened when you have a mismatch between the two? What happened when you have, well, for instance, when you're in your car and you read something. Some people can do that. I cannot, otherwise I vomit. And so actually you have about, why am I doing what I'm doing right now? And is it interesting for, people in companies? You have actually Google, Uber, or companies like that are working on vestibular visual integration, but because the goal is to have a car with a driver, and the goal is to be able to do other stuff, read, text, or while you are in the car, right?
Yeah.
But a big fraction of American population, or people in the world, they are motion sick. So how do you deal with that? So actually, it seems stupid, but from an economical point of view, it's actually kind of interesting for them if they can solve the problem. So who knows? Maybe next time you will interview me, I will work for Uber or Google.
We'll keep our fingers crossed. That would be great. Yeah. I'm curious, Guy. Just we know that you're also kind of a fan of MMA, and I'm just curious, just kind of quickly, as someone researching neuroscience, obviously knowing how delicate the brain is, I'm just kind of curious what you want to avoid head trauma. I'm sure.
I don't think about that. I'm just, sometimes humans are stupid, right? They like to fight, stuff like that. I just, well, at the same, well, I didn't say that I did judo for a while. So I think that's how I get interested very young to martial arts. And I don't like to fight. I just as a sport, I like, martial art. And I think MMA, even if some part of MMA, I don't like it. So for instance, I feel that when someone is knocked out, I think that the referee takes way too long to protect the guy that is already on the floor and obviously knock out.
Right.
That's the part that I think we need some optimization. I don't know, the guy gets a penalty if he punch the guy is already knocked out, obviously. And so this part I really don't like it, because you said, you didn't say it, but you wanted me to say that, yeah, that's a very dangerous sport. Yes. And you can have brain traumas. So my take on that is, I don't know why I like it.
Hey, I'm also a fan myself, and that's
Yeah, I think humans just have an inherent appreciation for violence and destruction, especially when you're not involved in it.
Yes, right. Especially when there's a cage separating you from it, so it can't
Oh,
affect
yeah,
you.
yeah, exactly. I think I also like. So recently, I was watching a lot. So following necessarily Francis Ngannou.
Ooh,
So he's the heavyweight UFC last champion.
he's a big
And
boy.
yeah,
That's
he's a
it.
big
That's
boy.
until I get my chance with him. I'm gonna take that belt back. No, I'm kidding.
Yeah, go ahead. I think I like the guy more for not well. He's the Mike Tyson of the MMA, right? He's a puncher like crazy.
Mm,
And but
yeah.
what I like about the guy is a story. So I don't know if you know his story, but he came from Africa,
From Nigeria, right?
from Nigeria,
Mm.
Or what was it?
no, not it's not Nigeria. It's isn't it Cameroon?
That sounds right.
I think it's Cameroon. And basically tried to go to Europe, crossing, the way people try to illegally come to Europe. And he tried I think 6 or 7 times to cross, to come to Europe. Then
Wow.
he arrived in France, and he was homeless for a
Wow.
while. And in this association for homeless, he met someone, and he was always talking about boxing. He was a big fan, but he didn't even have a TV. He was always talking about Mike Tyson and videos of Mike Tyson and so on. And he met someone that say, "Oh yeah, you should talk to this guy. He has a boxing club, and you should go there." And he went there. The guy saw Francis Ngannou and say, "Oh, you want to try?" He gave him some gloves and everything because he had nothing. He started to work, find a small job, but making money was he didn't have much money, nowhere to live to. And and then the trainer saw the guy, and he was naturally gifted. Obviously, he was punching very hard, even like naturally, right? And it started like that, and then he started to win some game. Then he started MMA already. He was already in France when he started in UFC, and now he's in the US with a very good trainer, very good team, and now he's a world champion. And he started 6 years ago.
Wow.
And so this guy is gifted. So basically, he was saying when he was younger, he was opening a door and he was breaking doors. Just opening a door. He has,
Right.
he's just strong, right? And I think, at least for this guy, I like MMA, but I like also for instance, in this case, the story beyond
Right.
believing in yourself and think that you can make it. And I think it's a good story, right?
I like it. I often have said that Joe and I are the Francis Ngannou of the podcast world with all of our. No, I'm kidding. I've never said. Joe, I'm tempted to kind of wrap things up on that note, but I wanted to give you a chance to add anything else if you wanted to.
Oh no, I just wanted to thank Guy for being on today, and also we got a chance to get lunch yesterday, and he showed me a great pad thai spot. So I've been thinking about that ever since. But yeah,
Sure. Come next Friday. Well, thanks so
gotta
much,
make
guys.
another
It was
trip.
very fun.
Well, of course, the pleasure is all ours. We kind of started taking this episodic approach maybe 2 weeks ago or something, and neuroscience was kind of a natural place for us to start because I think it ties in very well with themes that Joe and I talk about on the show all the time, whether it be persuasion or Cialdini's work on influence, etc. But also talk with people that are involved in research is important because another thing that Joe and I talk about is the power of small teams, the power of innovation, etc. And so having Guy here talking about how all those concepts come together in an important area of research and also in the research team was really exciting for us. Guy, we know that you are busy, or that you're a very busy person, so we appreciate you taking time to do this with us today. And we will definitely keep in touch. But until next time, I am Jimmy Hackett signing off for Joseph Stanford and Guy Bouvier saying ciao.